RNA. So we have 3 different kinds of RNA. We have the mRNA, and you don't have to write this down because this is review from yesterday. The M stands for messenger, right? Messenger RNA. This is the messenger in that it is produced from DNA and it travels from the nucleus out to the ribosome where a protein is made. Ribosomes contain the second type of DNA, our rRNA. Ribosomes are made up of rRNA. Sorry about that. And then finally, the third type of RNA is called tRNA. And if you look at the shape of this, it's kind of shaped like a lowercase t a little bit. tRNAs carry amino acids around with them. They transfer them. I'll show you how that happens today. Yes, Peter?
What does RNA stand for again?
Ribonucleic acid. Ribonucleic acid. So DNA is deoxyribonucleic acid. RNA is ribonucleic acid. So nothing necessarily you need to write down here. Just want to kind of give you some context where we were. So now transcription is the process, and like I said yesterday, if you want to just write down what I highlight, that's probably easier. In transcription, we have DNA serving as a template to produce RNA molecules, and specifically mRNA molecules. So just like what you saw in the packet earlier in the week with DNA replication, where DNA was getting copied, where we highlighted that all up, it's the same process. Instead of making DNA, though, we're making RNA. Same process, though. DNA unwinds, bases are added in that according to Chargaff's rules, that kind of thing. Let's talk about this word transcription. I asked you last night to think about what a transcript is. Anybody know what a transcript is? If you have older siblings, that are thinking about going to college, you might hear them talking about transcripts. Nobody? Go ahead, Mateo.
It's like something that's written down for, like something that was spoken and then it's written down.
Okay, and that happens in the medical field. Doctors sometimes take a digital voice recorder and record their patient notes, and then they have a medical transcriptionist write it down. But here in school, you have a transcript also. Has anybody heard that word with relation to education, with relation to colleges? Sean?
So it's like a record.
It is. You have your official school record. That contains your grades, your GPA, your absences, all that kind of thing. That's like DNA. It stays here at Lake Park. We don't want to send your official grade record out to one college, because then you only get to apply to one college. There's only one. You send that original copy out, there's only one copy to be had. Instead, what Lake Park does is we send copies of your official record, which are called transcripts, to the schools of your choosing, or the job of your choosing, or the branch of the military of your choosing. So transcripts are copies of official school records that get sent out. Much like we get copies made, mRNA copies, bless you, of our official DNA instruction manual, how to produce proteins. The DNA doesn't go out and do that. The DNA's got to stay in the nucleus because there's all kinds of different proteins that need to be produced. So we get transcripts made, copies of the different chunks of DNA, and then those mRNA copies go out and that's the set of instructions of how to make proteins. That's what the ribosome uses to produce the proteins. So that's where the word transcript comes into play. If you remember when I was using the different noises to talk about the difference between prokaryotes and eukaryotes. Remember that prokaryotes have no nucleus. So everything kind of takes place in the cytoplasm. We're going to be focusing our energies here today on eukaryotes that have a nucleus. So the RNA is produced in the nucleus, and then it moves out to the cytoplasm. So in eukaryotes, RNA is produced in the nucleus, and then the protein gets assembled out in the cytoplasm, which we'll go through in just a little while.
Wait, the prokaryotes have a cytoplasm or they don't have a cytoplasm?
They have a cytoplasm, they don't have a nucleus. Prokaryotes have no nucleus. Like I said, we're going to be focusing our attention on the eukaryotes, like us. What happens when the cells have nuclei? We need more time?
Yeah.
So transcription, since we're making RNA, the enzyme that's working as RNA polymerase, when we were making DNA during replication, it was DNA polymerase that was being used. Transcription uses RNA polymerase. Other than that, like I said with it when I pointed out the diagram from the packet, it's pretty much the same stages, the same steps.
Kind of sounds like polymerization.
Mm-hmm. That's exactly what it is. We're making a big, long chain of nucleotides. So step one, transcription in 3 easy steps, I believe was what I ended up with. Step one, the two strands of DNA are separated. DNA strands are separated. There, make it a shorter sentence there. So step one, DNA strands are separated, just like in replication. Step two, RNA polymerase is our enzyme. Uses one strand of DNA as a template to assemble RNA. Now there's one goofy difference here. We're working with creating RNA. So if our DNA is A, G, C, T, we're using that as a template to make RNA. Think about the base pairing rules, think about Chargaff's rules, think about the bases that are found in RNA. If there's an A in DNA, what RNA base is going to be put across from it?
T.
Not an RNA.
C.
U. There we go. U. That was one of the differences of RNA yesterday. Remember that we have uracil, that base pairs with adenine. So that's one important difference. Now in the other areas, so now G is going to pair up with C, C is going to pair up with G, and T will pair up with A. So all of that's the same. But if we have an A in DNA, then a U will be brought in to base pair with it. The U is complementary to the A. Bless you.
Thank you.
How does our RNA polymerase know when to start making mRNA? Regions of DNA that are called promoters have this code that tell RNA polymerase, bind here. This is where you start transcription. They have specific base sequences, and they're signals that show RNA polymerase where to begin making RNA. And then there are similar signals that tell RNA polymerase to stop. So RNA polymerase won't transcribe an entire chromosome. It's only transcribing one gene and turning that one gene at a time into a protein. So it has to know just to transcribe one little chunk of DNA. Furthermore, once that RNA transcript is made, this doesn't seem very efficient, it's not, but the thinking now is, the theory is that there are certain genes that, depending on how they are edited, can produce many different proteins from the same chunk of DNA, because it's only estimated that we have about 10,000 genes. Humans, complex humans, there's only about 10,000 genes that have been identified. How can an organism so complex be made from only 10,000 genes? Scientists think this is the key, that there are different chunks that are edited out depending on the protein that's been produced, or that's being produced. These areas that are cut out and discarded are called introns. So the same chunk of DNA can be edited in different ways to produce different proteins. The portions that are cut out and discarded are called introns. They are said to interfere. That's why they're called introns. They interfere with the actual code.
Is this DNA?
This is RNA.
Wait, okay, so the RNA edits.
So this is
itself?
after mRNA is transcribed, then it's edited before it goes out and gets turned into a protein. Sorry, that was unclear. Yeah, this is RNA editing. So introns are taken out while the mRNA is still in the nucleus. The rest of that mRNA that's going to be used to produce a protein, called exons. They're spliced or glued back together to form the final mRNA. And exons are called that. You can think they're going to exit the nucleus. They're called exons. Scientists call them exons because they are expressed. They're the ones that are sent out to the ribosome and used to produce a protein. this is what I was telling you about before. There's probably not anything you necessarily have to write down here. But again, if we have, this doesn't seem very efficient unless we think about if there are pre-mRNA molecules that can be cut and spliced in different ways, then you can have a single gene produce several different forms of RNA, which would then produce several different types of proteins. Introns and exons are also thought to play a role in evolution. Very small changes in DNA sequences can have dramatic effects on how genes affect cellular function. Again, not anything, we're going to get to evolution next semester, so it's nothing you need to worry about right now.